The views expressed on Skeptically Speaking are not necessarily the views of this station, its affiliates, sponsors, or advertisers.
Bring it on! The skeptics await your letter. Your call, tweet, homing, pigeon, whatever. Desiree Shell gonna raise a little hell, metaphorically that is. Cause we're skeptically speaking.
Hello and welcome to Skeptically Speaking. I'm your host, Desiree Shell. My guest is David Kroll, molecular pharmacologist and science communications director of the Nature Research Center at North Carolina's State Museum of Natural Sciences in Raleigh. Welcome back to the show, sir.
It's great to be here. Thank you so much for having me again. And congratulations. This is show number 150. So congratulations to you and the entire team for doing this on behalf of science.
Oh, we love it. We love you guys. Now today's show is all about fungi, but before we get to anything else, I'm going to have to demand that you actually share the story that you shared with me before the show.
Oh, this was a great story. So a wonderful segue into fungi is that they've been used for years for psychoactive purposes before they were even used for culinary purposes. And there's a specific form of Amanita fungus that produces a chemical called ibotenic acid that is a hallucinogen. But it turns out that when you take it and you digest it, it gets converted into another chemical called mucimol that is actually a better, more potent hallucinogen. And legend has it that in Siberia and among Russian royalty, that peasants would drink the urine of royalty who had been taking this stuff for hallucinogenic purposes because the urine actually produced better hallucinations than the original mushroom itself. And this was observed because of reindeer eating the urine in the snow of royalty who perhaps evacuated their bladder out of doors.
And I think we can all understand why I wanted to start the show with that story. Thanks, sir.
My pleasure. I'll be here all week. Thank you.
So maybe let's begin at the beginning. What is fungi?
So fungi are, and some people call them fungi, some people call them fungi. These are predominantly single-cell organisms that we now know are distinct from either plants and bacteria or animals. Fungi essentially evolved or diverged from plants and bacteria probably about 1 billion years ago. Most people think of fungi as mushrooms, which are actually the fruiting bodies of these microscopic organisms. Fungi are usually single cells that then form these long filaments in the soil predominantly of mats of these intertwined cells, almost like sponges, that play an important role in the carbon cycle of the planet by breaking down dead foliage, dead trees, any organic matter that's in the soil. So that's how they differ from other types of organisms. Now, what's different? Fungi are actually closer to us than they are to bacteria, even though they're microscopic. They are members of the eukaryotic family, meaning that they are their cells have nuclei. They have their DNA packaged in nuclei in these microscopic entities called nucleosomes, big batches of histone proteins around which DNA is wrapped. And so if you were to just look at a fungal cell, you would say that's much more like a animal cell than it is like a bacterial cell.
And fungi are actually the most diverse kingdom, correct?
They are. Genetically and morphologically, there are estimated to be 1.5 million species of fungi, and perhaps some of the least studied, because only about 5% of these 1.5 million have actually been formally classified. So there is a whole world out there that we still don't understand completely, particularly in our old growth forests that are decreasing in numbers around the world.
So what kind of species would fall in to the fungi category?
I would say, we first started off talking about Amanita, and these are fungi that have been used for hallucinogenic cultural purposes, all the way to series of Amanita species like Amanita phalloides that actually has a potentially fatal liver toxin that's inside of it. Most people are probably familiar with some of the culinary fungi like button mushrooms, like shiitake mushrooms, oyster mushrooms, and that sort of thing. But predominantly, most of the fungi that are of biological significance are those microscopic fungi that are in the soil. Probably one of the most common fungi that people might have heard of is called Aspergillus. Aspergillus is a fungus that grows on improperly stored grains, particularly peanuts, and is one of the kinds of fungi that are regulated by food and agricultural authorities, because it also produces a very potent liver toxin, such that grains that are contaminated with Aspergillus might actually cause severe adverse reactions in people eating, say, for example, contaminated peanut butter.
Convenient. Good.
Yes.
I have to say, one of the reasons that fungi is so interesting to me is because of the ways that they reproduce. Can you give us a brief primer on the sex lives of fungi?
Well, fungi actually can reproduce by themselves or with others, and I think fungal reproduction is perhaps one of the more interesting types of reproduction. They can reproduce asexually, that is, through simply cell division, just as bacteria do, where they simply divide, their own DNA divides, and it segregates to two cells that are clonal partners of one another. But as we know, sexual reproduction has its benefits, not just among humans, but most certainly among species that have been around for 1 billion years, and that is sexual reproduction allows a degree of genetic diversity and recombination that produce diverse offspring, if you will, that have different traits than the parents. And so fungi can reproduce sexually. These think about things like yeast. Yeast can actually mate and produce diploid or two genetic or chromosomal complements where one side is taken from one parent and the other taken from another.
There's also an amazing number of ways that fungus disseminates spores.
Spores are the kind of resting form of fungi, where the cells, particularly in conditions where water is not available, or when they reach their final maturation in fruiting bodies, produce these just really amazing mobile forms of themselves. We think about the fungi growing in the soil, but when they get to this spore form, where an entire top of a fruiting body of mushrooms can essentially puff out and disperse these microscopic, hard, circular versions of their former selves into the air and literally get carried feet to miles. It's a really remarkable way for a stationary organism to be able to travel long distances. I
found it really interesting that some of them actually use natural water pressure. They squirt, really.
Yeah. So there are some fungi. I didn't know this till I started studying fungi, but there are some that actually build up pressure inside of them, and as the outside of the top of the fungus can actually starts to degrade, the pressure from inside, dare I say, it's kind of like a pimple bursting, that the pressure from inside causes a forced ejection of spores rather than them just being kind of passively dispersed. I think most people have come across puffball mushrooms. If you're out hiking and you kick one, then you see this big fluff of spores. Well, some of these other mushrooms produce these, they call them ballistospores. That is ballistic ejection of fungal spores.
Well, actually, there was a report in the journal PLOS ONE that fungi may be one of the fastest land species, and they clock speeds of up to 88 kilometers per hour and produce accelerations 180,000 times greater than gravity.
You'll have to send me that paper. That's fascinating, but entirely not surprising.
You're listening to Skeptically Speaking. I'm here with David Kroll, natural products pharmacologist. David writes at the Terra Sigillata Pharmacology blog and less formally at Take As Directed. He's also a monthly contributor to Science-Based Medicine. So there are a whole bunch of examples of how fungi has been useful to us personally, starting with alcohol.
Yes, the yeast Saccharomyces cerevisiae is actually named for the very process that it's involved in. Saccharomyces standing for the metabolism of sugars, and cerevisiae, those of you who study Spanish know that cerveza is the Spanish word for beer. And in fact, going back probably to the Middle East, I believe Egypt, about at least 4,000, possibly 6,000 years ago, it was observed that grains that had been stored and cooked to release the sugars in them could spontaneously be converted into a bubbling mass that produces the short-chain alcohol, ethanol, that we all call alcohol. And that process is called fermentation, and obviously by experiencing the euphoric, intoxicating effects of ethanol, we as a species decided to partner with yeast and produce large amounts of a variety of fermented beverages.
Of course we did.
You know, and what's really interesting is you sort of step back and you say, why did yeast develop, this particular type of process? It takes a lot of energy to convert sugar into ethanol, and it's actually a biological defense strategy. We'll probably talk later about the different chemicals that fungi make to defend themselves against other organisms, and in fact, most bacteria that would be competing in the soil or in other organic matter for nutrients can't tolerate as much alcohol as yeast can. And so the yeast production of alcohol is actually to kill off other competing organisms that might be trying to latch on to the sugars and other goodies that are in the soil that Saccharomyces cerevisiae might be wanting to use.
So is that the same process that makes bread rise then?
Exactly. The process of fermentation, that is the metabolism of glucose into ethanol, can also produce carbon dioxide. And for anybody out there who may be a home brewer, I know we've got a lot of scientists and regular folks who have home brewing kits. The first thing you see, first evidence of yeast fermentation is the bubbling or production of carbon dioxide bubbles. And in beer brewing, that's blown off over the top of the liquid. But think about this. You go ahead and mix yeast and some sugar in the context of bread flour, and you make a dough. Well, where's that gas going to go? The gas starts to form carbon dioxide pockets that give the bread its fluffy character. And based upon, if you use high gluten flour or so forth, you can get very small bubbles to very large bubbles. This is actually a form of biotechnology. This is where we've, as humans, where we've been using the life of the fungal kingdom to produce better products for us. And bread and alcoholic beverages are probably the two hallmarks of fungal contributions to our lives.
We have an email question from Leroy.
If you could meet the first guy who said, "Hey, I'm going to eat this weird puffy thing growing under this rotten log," what would you say to him?
It would really depend on what that puffy thing looked like. But you can imagine why people would have eaten the fruiting bodies of fungi out in the wild if you had nothing else to eat. Think about this as you're walking through a forest and there are all these sticks and leaves and pine needles, and then you see this big 10-centimeter wide lush white beautiful thing that looks like you could, cook it up, and of course you'd want to eat it. And so it seems very likely that that fungi were eaten by people for many, many years, simply because they seem to be the most palatable of anything available in the forest.
Well, I wonder how many people had to die, really, to figure out which ones were good to eat and which ones were not, because there are various different properties that fungi have.
Yeah,
some make you high, some will sate your hunger, and some will kill you.
There's a Croatian proverb that says that all mushrooms are edible, some only once. and that's because mushrooms as mushrooms and other fungi as biological defense mechanisms produce metabolites that are toxic to other potential predators, whether they be microbiological predators like bacteria or other fungi, or whether they be mammalian predators like truffle-digging pigs and humans. And what's really surprising to me when I started looking at the biological effects of fungi, I'm not a big mushroom eater, but I've been interested in fungi as a kind of an intellectual pursuit, is that they produce a lot of toxins that do nothing to each other until they are metabolized in the liver to something that is extremely toxic. You think about it for a second. Fungi don't have livers. Plants don't have livers. However, mammals do. And the fact that we can convert these fungal products into things that will destroy our livers or cause toxicity in our brain, or maybe not toxicity, but at least cause hallucinations or unpleasant experiences, cause our gastrointestinal tract to behave in unpleasant manners, kind of tells you that fungi develop these compounds not so much for themselves as they were to defend themselves.
We have a question from Curious.
We know that some species of fungi produce hallucinogenic substances. What are the functions of these chemicals in the fungi themselves?
Sai's asking a question that I think all people in natural products ask, and that is, why do organisms devote all this effort to making these extremely complex molecules, and again, molecules that bind very specifically to receptors in our brains or the brains of grazing animals? To be honest, my most educated guess is that they produce these things to cause unpleasant effects in other organisms, hoping that we won't eat them again. But on the other hand, one could say that, and this was a theory put forth by Michael Pollan, the food and environmental writer, in his book The Botany of Desire, where he posits that a lot of plants and fungi that we have, that we gain psychological benefit from psychotropic or brain mind-altering substances, we actually that provides an evolutionary advantage because other organisms will actively cultivate them, as we do with things like marijuana and psilocybe type hallucinogenic mushrooms. Not we, you and I, Desiree,
right? Of course not.
But other human beings, right, on the planet.
And of course, now that we've started talking about hallucinogens, here's a question from Jenny.
Is there more than one kind of hallucinogenic mushroom?
Probably the most common is psilocybe cubensis. People call them magic mushrooms. But there are multiple mushrooms that produce psychoactive substances. A lot of them act on these serotonin receptors that Curious studies, these serotonin 5-HT2 receptors. But we have mushrooms that produce other types of compounds that work on acetylcholine receptors in our brains, or muscarinic receptors. That is, these are fungi, maybe not so much mushrooms, but the microscopic fungi that produce compounds like muscarine, which is an old-fashioned, old-timey, if you will, hallucinogen. No, I would say that fungi probably produce some of the most diverse natural hallucinogens out there. So when one's looking for hallucinogen, you turn to fungi.
Well, now I'm wondering, when I was doing research for this interview, I came across a lot of this is how you can tell a poisonous mushroom from not a poisonous mushroom. Do those kind of rules actually exist?
Oh boy, this is
things that sort of span across mushroomhood.
Yeah, certainly, and there are local citizen science societies, mycological societies that help to train one another. What is a edible mushroom versus what is an inedible mushroom. And again, I said they're all edible, some only once. There are some general rules looking at pictures of mushrooms to know which family members might be edible and which ones you need to absolutely stay away from. But the morphology and the coloration of mushrooms varies based upon where they're growing. And I think that picking wild mushrooms and eating them without anyone who is a very highly trained scientist or who has a portable polymerase chain reaction machine to do DNA analysis, you probably want to stay away from mushrooms out in the wild. Even very experienced mushroom pickers will have experienced adverse reactions from mushrooms, maybe not dying, but certainly having effects in their gastrointestinal tract that make them wish they were dead.
That was clear. Thank you. So, speaking of death, when I was looking at the myriad articles for this show, I came across one in the Daily Mail. Yes, I know. And it attributed the death of Brittany Murphy. That was the actress from the movie Girl Interrupted, the one that wasn't Angelina Jolie or Winona Ryder, that one?
Yes.
To a fungus-based illness. So I sent you that article, David. So what do you think?
Yeah, that blew me away. I had no idea that this had occurred with this actress. And it turns out that what was infecting her home was this toxic black mold. The British spell it M-O-U-L-D. Do you spell it M-O-U-L-D in Canada?
Probably.
Mold?
I spell all words wrong. That's what spell check for is. Sorry, honestly.
But so the toxic black mold, that's the one that you hear being the cause of folks needing to remediate buildings, old buildings, particularly in more humid environments, cellars, any place that gains a lot of humidity without a lot of airflow. It turns out that several species of fungi, in this case it was this Stachybotrys chartarum, form of the black mold, produces toxins that when airborne can actually cause really severe and specific inflammation of the lungs and actually hemorrhage of lung tissue, and so pneumonia and hemorrhagic lung disorders are actually well known to be associated with the toxins produced by this Stachybotrys, which is simply, it's fascinating. This idea that mold can cause severe life-threatening illnesses, I think, is something that we've only begun to appreciate medically in the last, 10 or 15 years.
Really? The Daily Mail wrote something that wasn't actually hyperbolic? I'm kind of confused.
The Daily Mail, that was, I wouldn't say that, that it was highly scientifically accurate, but the reporting and the conclusions that were made were actually true and not unnecessarily inflammatory or sensationalized. The Stachybotrys produces these compounds that we keep isolating in the laboratory, called trichothecenes, and the one that's in this black toxic mold is called satratoxin, and there's a whole bunch of these satratoxins, A, B, C, D, essentially the order in which they're discovered, they get a letter at the end of them. But yeah, these are really potent compounds that kill cells or can cause severe inflammatory reactions, almost like autoimmune reactions, and they occur, when you inhale the mold. It occurs in the lungs, and there's a whole series of scientific literature out there where these satratoxins and related atrotoxins will actually destroy the cells in your nose, the olfactory cells, the neurons that are responsible for converting chemical signals into smells that your brain processes. So these molds are not, these particular molds are not very desirable.
And we'll be right back with more on fungi, the good stuff about fungi, with David Kroll, Science Communications Director at the Nature Research Center at North Carolina's State Museum of Natural Sciences in Raleigh, after this.
Skeptically Speaking wants you to question everything. To be a part of the discussion, call and record your question on our listener question line at Speak 73101. That's 773-257-3101. Long-distance charges may apply. Sunday nights at 6 Mountain Time, join our Ustream chat room to listen and ask questions as we record interviews live. Just click the Listen Online button at skepticallyspeaking.com. You can also email your questions and comments to live@skepticallyspeaking.com, or if you're on Twitter, tweet us at Skeptical Radio. To find out where Skeptically Speaking airs near you, to listen to past episodes, or download the weekly podcast, check out our website at skepticallyspeaking.com. While you're there, follow the links to connect with us on our Facebook page, our Twitter feed, or to subscribe to the show in iTunes. And now, back to the show.
Welcome back to Skeptically Speaking. I'm your host, Desiree Schell. Today I'm talking to David Kroll about the weird, wonderful world of fungi. Until very recently, David was a molecular pharmacologist where he focused on the discovery of anti-cancer drugs from filamentous fungi. Now he's the Science Communications Director of the Nature Research Center at North Carolina's State Museum of Natural Sciences in Raleigh. So let's talk about some of the wonderful things that fungus does. Now, different types of fungi have been used throughout history to cure illness and disease, right?
Yeah, fungi. It's really interesting. The just like plants, which are a source for literally hundreds of medicines that we use. Fungi also produce chemicals that we have harnessed to become medicines. I don't know exactly why, but a lot of this work was originally done in Japan, and I think there's a strong culture of microbiology research. Pun not intended, a strong culture of microbiology research that extends specifically to fungi. And probably the most famous fungal-derived drug that listeners will know are the statin class of cholesterol-lowering drugs. So things like lovastatin and this whole variety of chemicals. And they were originally discovered by this Japanese scientist named Akira Endo back in the early 1970s. I think the first paper was 1971. And he actually had generated or developed a respect for mushrooms from his grandfather living in some rural areas of the main Japanese island. And as he became a scientist, he became interested in these chemicals that fungi made. And we talked before about the reasons that organisms will go through the energy of making these chemicals to protect themselves from other organisms. Well, it turns out that fungi require certain forms of cholesterol for their cell membranes. Okay, so even though fungi also have cell walls, the cell membrane is really important. And unlike us humans, there are a lot of different kinds of cholesterol molecules in fungal cell membranes. Well, it turns out that fungi make inhibitors of cholesterol synthesis that they use to kill fellow fungi. And Endo discovered this and started actually working on developing this particular class of chemicals to treat high levels of cholesterol that predispose people to heart attacks and strokes. I mean, it's really fascinating.
Well, and actually you were working on something just recently involving cancer. Tell us about your research. Until
I left the university and moved over to the Natural Science Museum in Raleigh, I've been working on a National Cancer Institute, U.S. National Cancer Institute project to identify anti-cancer chemicals from a specific library of fungi that are housed by a colleague of mine named Cedric Pierce, who has a small company called Mycosynthetics in Hillsborough, North Carolina. And he had inherited a fungal collection from an old mycologist named Barry Katz, who in the 1980s started collecting fungi from decaying plant matter and soil from all over the world, to the point that Cedric now has 55,000 cultures of these fungi. And together with my other buddy, Nick Oberlees, who's over at University of North Carolina at Greensboro, Cedric's been growing up these fungi. Nick his laboratory's been extracting out the chemicals that are made in the fungi, and then we were testing them in cell culture for anti-cancer activity. And it's really fascinating because these fungi produce kind of a chemical diversity. That is, they produce a great combination or a great spectrum of different shaped chemical structures that can't be easily man-made. And so they allow us to test compounds against cancer cells or any other kind of biological activity with essentially chemicals that can't be duplicated or can't easily be duplicated by man or woman. That would be.
And actually, fungi plays a role in some multiple sclerosis medication as well?
We tend to think about these natural products, naturally produced chemicals like morphine and some toxins like the muscle relaxant succinylcholine that's used in surgery. We tend to think of these things as really old chemicals, but it turns out that a compound that was just approved in the U.S., and I believe it's approved in Canada, to treat multiple sclerosis, it goes by the name fingolimod, was just approved late last year, and it turns out to be a slightly modified fungal metabolite. Comes from a chemical called myriocin, myricin, which is a fungal metabolite that interacts with nerve cells in a totally previously unappreciated manner to reduce the muscle damage, the nerve damage in multiple sclerosis.
And maybe this is going to be a silly question, but I wonder, how does a researcher get the idea to look at certain things like fungus for these very specific applications?
I think part of the reason is that point I made before about the chemical diversity. I would say in the 90s, drug companies started and chemists started saying, well, we're going to make these huge collections of chemicals that we can make in the laboratory, and we can make 5 million different ones, and we're totally going to create cures for cancer and so forth. And what really happened out of these groups of chemicals, they're in the business, they're called combinatorial libraries. In these combinatorial libraries, there essentially wasn't a lot of diversity. So the same compounds were being isolated by the same drug companies, and no one could really get good patent position on producing drugs from these combinatorial libraries. And so after all this spouting forth about medicinal chemistry, it turns out that the best medicinal chemists have been operating for millennia out in nature, and those are plants and bacteria and fungi and marine creatures. All these organisms have been making chemicals of different shapes and sizes that are probably poisons at high enough doses, but when we use them at smaller doses, they can act very specifically with molecules in our body that mediate disease processes. And so for that very reason, I think you'll see particularly a lot of academic researchers, university researchers, nonprofit research institutes being really interested in these naturally derived compounds because, it takes a lot more work to work with these natural compounds, but the payoff is really so much greater.
You're listening to Skeptically Speaking, and I'm talking with David Kroll, Science Communications Director of the Nature Research Center at North Carolina's State Museum of Natural Sciences in Raleigh. So it's not just medications that fungus is working on, let's say. We're figuring out different ways to use it all the time. There was a Scientific American article recently about a fungus that protects rice from climate change. Apparently it works through, and I'm going to mangle this again, endophic fungi?
Oh, endophytic fungi, right.
Mangled.
These are fungi that actually, they grow on the roots of plants, and it's kind of a symbiotic relationship where the fungi, they make chemicals that are used by the plant themselves, in many cases as nutrients, and in other cases as defense mechanisms for the plants.
Well, I think that was the point, is that the fungus was actually protecting the rice from climate change.
Fantastic. By making the rice more responsive to changes in the environment.
Going through the research for this, I'm thinking fungi does everything now. Like, it's almost magic. We're using it to remove stains. Mm-hmm. We're using it to detoxify polluted soil and water. Mm-hmm. And we're looking at using it to produce biofuel.
I mean, this is amazing. And I have to tell listeners that when you're done listening to us, go to your computer and search for a TED Talk by this mycologist named Paul Stamets, Stamets. He has this beautiful 18-minute talk with gorgeous pictures, essentially giving 6 ways that fungi can save the planet. And Des, you're talking exactly about these issues that Stamets is, and probably the most relevant to us today is the fact that fungi, by nature of their ability to decompose complex decaying products from trees and plants, those very enzymes that they use to break cellulose, tree bark, that kind of stuff, into ultimately sugars that can be metabolized and used by other organisms, is the exact same process that can allow us to make biofuel ethanol from cellulosic products. That's been the biggest hurdle in truly making biofuels a sustainable, replenishable resource, that is the changing or the modifying of cellulose into sugars. And Stamets shows in this talk this beautiful picture of corn cobs, that is not corn with the hulls still on them that contain the corn starches, but just thrown away corn cobs being overtaken by fungi and producing these large mushrooms, essentially showing that we can take single sugars, or we can make single simple sugars from cellulose that we can't digest, but fungi can digest, and then we could use those sugars to ferment again with other fungi to make ethanol for biofuel. It's amazing. It really is. And just making it cost effective is what a lot of companies around the world are trying to do right now. And there's some great pilot plants down here in North Carolina at a company called Novozymes, which is the American offshoot of the big Dutch company Novo Nordisk that makes clotting factors for hemophiliacs. You're gonna see fungi playing a huge role in the future of sustainable energy.
Are you telling people to invest in fungi?
Invest in fungi. Buy low, sell high.
All right, we have a bunch of audience questions, and I'm going to try to get to a few of them. This is from Twitter, from Ev Luke.
What are slime molds, and how are they different from fungi?
Yeah, so slime molds are, they look as they sound. They look like slime. And these kind of gelatinous type organisms, they'll also grow on bark and on the soil. There's actually, I see it around here a lot. There's this slime mold that's called the dog vomit slime. And even though I have a beagle in the house, the dog vomit slime is not dog vomit, but it actually is a slime mold. And we call them molds, but it's actually a misnomer. These were formerly thought to be fungi, but upon genetic analysis, they belong to the family of protists. So they're no longer part of the fungal kingdom, but they do share characteristics with fungi in that they use they can reproduce by sporulation, that is making these spores. But I mean, from a functional standpoint, slime molds are serve kind of the same purposes in nature that fungi do, in the sense that they decay soil, they decay fallen trees and so forth. But semantically and genetically, they are no longer considered to be fungi.
And from the chat, from Baitor.
If they weren't so dirty and dusty, would slime molds be edible like mushrooms? Or would they be poisonous and hallucinogenic?
Oh, that's a good question. I am not sure if any slime molds have been known to produce hallucinogens. That's probably the one where you'd look at it and say, here is this thing on the ground that looks like dog vomit. I definitely am not going to try eating that. No, to be honest with you, with my and I don't study slime molds myself, but I've not come across any information as to whether they produce toxic secondary metabolites or hallucinogens.
Okay, now we have serious questions here, but there seems to be mushrooms that smell like death. So tell me about that.
Yeah, these are, and we have these throughout the plant kingdom as well. There are organisms that will produce chemicals that make them smell like decaying animal matter or dead animals. And they produce, the predominant chemicals they make are appropriately named things like putrescine. Putrescine is, we make them, and they are produced, these they're called polyamines. These polyamines are made in high concentrations in decaying flesh. So you're asking, Desiree, why on earth would a plant or a fungus make something that smells like decaying flesh? Well, it turns out that it attracts insect activity and attracts predator activity, so animals like vultures. And by eating the fungal or plant material, this is a way to also spread genetic material that is either spores or these microscopic forms of the fungi called hyphae. So it's actually, while it may smell putrid to us, this is actually a genetic strategy by the organism to propagate itself and propagate itself over long distances.
Evolutionarily, that is remarkable.
It just blows me away. I mean, we tend to think that we're so smart, but these organisms and unicellular organisms, they've been doing this for years, for millennia of millennia. This is where biology is just wonderfully exciting. I mean, you could spend your whole life studying this, and I have.
Oh, wait. Here's a question from the chat from Jeff.
I live in West Virginia, up above the Ohio River, and there were many different types of mushrooms and toadstools that popped up over the yard with cigar trees, black walnut, and pine trees. Is there a reason why last October-November was so productive with fruiting fungi?
Boy, I've heard that across the Northeast, and I guess you would still consider West Virginia to be Northeast or at least Central East. Yeah, something about the combination of temperature, heat during the summer, and then temperature during the fall. Folks I know who pick mushrooms said that last year was an incredibly rich year for seeing a lot of mushrooms that they hadn't seen before, or in areas where they've seen mushrooms before, that there was just a tremendously large number. And this is the kind of thing that if you're a true mycologist, you might be able to predict when the weather might give you huge blooms of these fruiting bodies. But I think the science of knowing when mushrooms are going to fruit, I think is really a part of science that's not well worked out. But yeah, you'll see this kind of thing from season to season.
I am going to ask you a question that we may have to edit out, because it's from Cy Curious.
Awesome.
Some interpretations of witches riding broomsticks have postulated that the witches were really riding the broomsticks, which were coated in hallucinogens. Is there evidence for this? And can hallucinogens be absorbed through the vaginal wall in this manner?
Yeah, this is a great, great story. I mean, I get this question a lot about at Halloween time, and I've actually written about it. That in the potions that witches would make, there were a variety of hallucinogenic plants, not mushrooms, but hallucinogenic plants that were used to create for the witches when they ingested them hallucinogenic experiences that often included themes of flying through the air. And I think that's where the issue of witches riding on broomsticks came from. But the broomstick, David, you ask why. The broomstick was a way for the witches to rub the potions on the outside of their vaginas to get better absorption of the hallucinogens. This is kind of a similar reason to why people today will inject drugs. A lot of potions or hallucinogens that are taken orally, they first have to pass, they get absorbed in the intestines and they have to pass through the liver before they go to the rest of the body. And the liver is kind of like the catalytic converter of the body. It will chemically change stuff that you ingest into other chemicals, and in many cases that'll terminate the biological activity. But there are other ways that you can take drugs. One of them is under the tongue. We have a lot of drugs that are taken under the tongue that won't go through the liver, the first pass through the body. And then also the vagina and rectal drug administration can also be ways of getting chemicals into the bloodstream by bypassing liver metabolism. So it actually is a very serious question, and the combination of using hallucinogenic plants in witch potions, applying them to the vaginal area with a broomstick, literally, is the sort of folk reason that witches are portrayed as riding broomsticks.
So you've blown my mind, and and I can't really top that question with any other questions. So,
Yeah.
David, thank you so much for being on the show.
Thanks for having me. It's always a pleasure being with you folks.
We've included links to David Kroll on our website at skepticallyspeaking.com. And we'll be right back with more after this.
On the next episode of Skeptically Speaking, we're looking at the amazing abilities and potential of the human body. Evolutionary neurobiologist Mark Changizi joins us to talk about his book, The Vision Revolution, which looks at the evolution of vision from a novel new direction. And Dr. Tora Katsur discusses practical ways that science and technology can get us closer to the extraordinary abilities we see in science fiction. That's next week on Skeptically Speaking, on your local radio station, or online at skepticallyspeaking.com.
I'm here with Sarah Mathews, Sargent Fellow and a Principal Investigator at the Arnold Arboretum of Harvard University. She's interested in plant phylogenetics and in the question of how changes in light-sensing systems have influenced the ability of plants to survive and diversify. Thanks for being here, Sarah.
Great to talk with you.
Now, a great deal of your work hinges on the concept of living fossils. So can you explain that term?
Basically, what we see, cycads are one example, coelacanth, these fish, horseshoe crabs, are organisms that, if you look in the fossil record and compare those fossil species with what lives today and we can see today, they don't appear to have changed much. They look very similar to one another. And so that's where that term living fossil comes from. It actually was coined by Charles Darwin.
Now, the problem is that term is a bit misleading, isn't it?
It certainly has turned out to be in many cases. So the question of why are things so similar when they are so old, what explains that? Are they really younger than we think? Or for some reason have they not been induced to change over time as many other organisms have?
Well, why exactly are living fossils so important?
Well, I think that they're certainly interesting. They present us with an evolutionary puzzle. We don't really understand what's going on when things don't change morphologically. There are a couple of different models. One might suggest that some sort of stabilizing selection is sort of pushing from both sides of the spectrum to which they could explore, keeping them sort of centered on a certain morphology. Another model would say, well, organisms that have lived through various kinds of major changes in the environment might have been the most successful organisms at living through those major perturbations might be more likely to persist through ongoing and subsequent environmental change. And so that basically is the idea that early on there may have been selection for what we would call generalists who can survive without actually having to change a lot.
So really the idea is that living fossils can teach us a lot about the evolution of very specific species.
Well, the question is really what sort of speciation has occurred when something looks like it hasn't changed much over time. Is it really the case that it has really persisted since, in the case of cycads, before the dinosaurs existed? Or was there a lot of species turnover? Did a lot of things go extinct, but the few lineages that survived did survive, and then some of them, in turn, found a way to become very successful much later in time.
So how does one figure that out then?
Well, we can reconstruct a genealogy of all the species that are living today using information in their DNA sequences. And we can then take that genealogy, which is basically, you could imagine yourself walking along a road and there was a split in the road and one branch went one way and one branch went the other way, and that led to two different species. And for each type of split like that in the tree, we can use information from the fossil record to infer when that split occurred. So we know basically how old species actually are, or we can infer an estimated age for the age of the species based on this history in their DNA combined with information in the fossil record that gives us a way to put dates on the splits.
Now this is what you've been researching, correct, in cycads?
We did that in cycads, yes. It's really part of a larger project which is studying seed plants that's funded by the National Science Foundation. They're assembling the Tree of Life program, trying to really put together the puzzle of how these different groups are related to one another. And so the cycad study really is a part of this larger study to try and understand the history of seed plants themselves.
Well, how did you figure out that cycads weren't living fossils per se?
Well, we took our approach of asking if you can envision that you have all the species of cycads connected to each other via sort of these branches, just like a genealogy. We can ask if a single species is on a single branch, how far back in time does that branch connect to the branch of another species? If it goes very deep in time, that suggests that those species really are very old and would truly be called living fossils. If you take that one branch with that one species on it and trace it back in time and find that it connects to another species only 10 million years ago, that suggests that the species is young. And that's basically what we did using our DNA phylogeny. And we know that there's good evidence that DNA evolves at a certain rate, and so we can calibrate our tree. Basically, we can infer a relative rate of DNA evolution just by asking how closely related the species are, and then we can put that in a time framework by using information in the fossils themselves.
So what is the story of modern cycads then? When did they appear?
Just about 10 to 12 million years ago, which was in a period of time that we call the Miocene. And it was a time when basically on a global scale, climates were cooling and there was some drying occurring as well. This sea that no longer exists as our continents are currently configured was closed up at this time, and a number of currents that were circumglobal in the ocean changed basically, and that also led to changing climates. And so the cycads, these species that are fairly young, they all seem to have originated around this period of time when climates were cooling and drying, and they're becoming more seasonal.
So then how much has changed? Without the actual DNA evidence, would we be able to tell any difference?
The thing that's very intriguing is that although about 11 lineages of cycads persisted through 300 million years or so, not all of those lineages that survived are now become species rich. They weren't successful in the Miocene. So you could say that one genus of cycads became very species rich, and the genus that it was most closely related to sat there and did nothing. There's only a single species in that genus. So what's going on there? Why was one lineage very successful and the other most closely related thing? So you would think it would have the same capacity to diversify. Why did it not diversify? And so I think that there are clues in these kinds of comparisons that can help us detect, what was it? Are we just seeing? Are we not seeing? Is there a sort of cryptic variation there that we're not seeing? So, do plants really have the ability to change in ways that are much harder to detect than just by looking at their overall form, which is, of course, the only thing we can see in the fossil record? So, does their physiology change? And do some groups have more ability and flexibility to explore different kinds of physiological adaptations? And in fact, there are some interesting aspects of zamia, which a lineage of cycads that occurs in Mexico and South America and Central America, and it has developed a variety of different kinds of photosynthesis that we typically associate only with things like cacti and certain flowering plants in more extreme environments. So there does seem to be some physiological exploration, if you will, going on in some of the genera. Another thing that may be going on is that some of the genera may have much more variability in their chromosome structure. So at the level of the chromosome, some of them seem to be more variability. So maybe having a greater ability to change the structure of the chromosomes themselves may facilitate what we would call reproductive isolation, where within a population all of a sudden certain subsets of that population can't mate anymore, and so they become reproductively isolated and eventually diverge into separate species.
So would this be the case with all living fossils then? Or is this just plants we're talking about?
I don't know the other groups well enough. Certainly in some of the other groups that we call living fossils, and in fact even in some cases plants, so ginkgo biloba, the maidenhair tree, has also been called a living fossil. That's a case where diversification hasn't happened, and I think in some of the animal lineages that have been called living fossils, there also has not been this, what we would call re-radiation at a later point in history.
So does it matter if they're living fossils or not, ultimately? Like, will this...
It really doesn't. Okay. It really doesn't. I think only to the extent that it motivates questions about, what is the basic biology underlying their ability to persist and then their ability to rediversify, because it has implications for how we understand what's going to happen in the future. Are organisms going to be able to adapt to changing environments, and what sorts of expectations can we have along those lines?
Well, I think the ability to predict is a worthy endeavor.
It is, and I think we've got a long way to go to really understand. And then once we can understand, it's hard to know how that's actually going to help us, mitigate. But it should. It might tell us a lot about what sorts of structures and habitats and population sizes and so on are really important as a basic requirements for the persistence of ecosystems.
Sarah, thanks very much for being here.
Thank you. It's been fun to talk with you.
You can find links to Sarah Mathews' work on our site at skepticallyspeaking.com. Since you're there, maybe do click the links to our Facebook page, our Twitter feed, or to Google Plus to get program updates, and also to iTunes, where you can subscribe to the show, listen to past episodes, or leave what I can only assume will be a glowing review. Thanks for listening, and we'll see you next week on Skeptically Speaking.
Bring it on, the skeptics await your letter. Your call, tweet, homing, pigeon, whatever. Desiree Shell gonna raise a little hell, metaphorically that is. 'Cause we're skeptically speaking.
Skeptically Speaking is produced with the support of CJSR 88.5 FM in Edmonton, Alberta, Canada. Our studio assistants are Brent Kelly and Rochelle Saunders. Ryan Bromsgrove is our studio technician and research assistant. The show is produced and edited by K.O. Myers. Our theme song was written and recorded by A.O.K. And I'm your host, Desiree Schell.